Okay, my name is Sergio. I am a PhD student. Nahuel is my director, and Fernando is my co-director of my thesis. I'm from Colombia, not from Argentina. I live in a region that is very tropical, a lot of rainfall, but the precipitation, like events, they are not very frequent. Like it rains 100 millimeters, and there is 3 weeks no rain. So the commercial companies, they sell a lot of biostimulants, I don't know, like foliar fertilization, and there is a myth about that. So I want to ask you if first there is a little information about the use of those products and the effect on yield in those kind of environments. And for example, they say that the foliar fertilization using biostimulants, like the 30% of the yield can increase. And I know I want to know if there is a real scientific information about that, no commercial information.
Yes, there is scientific information, but one difficulty is that most of the scientific information that we have is about seaweed extracts or humic acids. When you are a scientist, you want to be able to characterize well what you apply to the plant. You want to be able to discuss the effects and to relate the effects with the composition of the product, maybe to build up a really scientific narrative. A problem is that many products that are on the market are in fact mixtures of many things, including some micronutrients, which makes it very difficult to interpret.
El micrófono lo más cerca de la boca posible, así se escucha.
No hubo sesión.
Sergio.
All the family here. Sí, estamos arrancando. Con consulta acá de...
Hacela en inglés.
Si no, sí, si te animas en
inglés?
Sí.
Okay. Sorry. Okay, my name is Sergio. I am a PhD student. Nahuel is my director, and Fernando is my co-director of my thesis. I'm from Colombia, not from Argentina. I live in a region that is very tropical, a lot of rainfall, but the precipitation, like events, they are not very frequent. Like it rains 100 millimeters, and there is 3 weeks no rain. So the commercial companies, they sell a lot of biostimulants, I don't know, like foliar fertilization, and there is a myth about that. So I want to ask you if first there is a little information about the use of those products and the effect on yield in those kind of environments. And for example, they say that the foliar fertilization using biostimulants, like the 30% of the yield can increase. And I know I want to know if there is a real scientific information about that, no commercial information.
Yes, there is scientific information, but one difficulty is that most of the scientific information that we have is about seaweed extracts or humic acids. When you are a scientist, you want to be able to characterize well what you apply to the plant. You want to be able to discuss the effects and to relate the effects with the composition of the product, maybe to build up a really scientific narrative. A problem is that many products that are on the market are in fact mixtures of many things, including some micronutrients, which makes it very difficult to interpret. And there is a gap between the scientific literature and the data on the field from companies. So, but I think that one thing is that if a company tells you, okay, we believe that this helps preparing the plant for the next drought episode on this, via this foliar application, etc. Show me the data. Where are the data? They should have the data. And any claim should be supported by data, not just by, okay, it's not written in the sky. There must be some information. So I think it's important that the grower or the PhD student making research on a topic challenges companies, because they do have data sometimes, but they have to share. They have to be more transparent. I think that's fair. They want they want the grower buy new products, new innovation, adopt new innovation, but okay, that must be fair, transparent, and it is still complicated. In order to make this technology efficient, workable, we definitely need more data, as you said also. The problem is that you will not find the last say, I would say, in the scientific literature. Scientific papers will tell you much about the potential effects of these products, but at the end, you need the data for this product in this condition on that crop. And the experience is that it's really case by case. If you take seaweed extracts, I could trace in the literature the use of one specific seaweed extract from one of the big companies in the field. For drought tolerance in citrus and in almonds. No, it was in vine, that one, comparing. And in one case, conclusion is, okay, you have better efficiency when applying on the soil. And the other paper, it was exactly the opposite, better efficiency when applied on the leaves, foliar application. Okay, that's the evidence, which means that it's in fact very difficult to generalize. Many times people ask me, is it better to apply in the soil or in the leaves? It's impossible to answer that question. You have to test. Although there are some meta-analysis, which I showed this morning, showing, okay, statistically, soil applications seem to be more efficient than foliar application, but that's only statistics, of course. For your individual case, it might be seed treatment, maybe. And so you really need to collect the data. The point is that it's important to agree on the protocols, on the quality of the data, on the wording that you use. We have to understand each other when talking about these kind of things. So that's why it's important maybe to have a regulation fixing the use of the words so that we can understand each other, and that we can also maybe adopt some standardized protocols for testing drought tolerance, for instance, how to do that. In Europe, we had a discussion about what kind of data do you need to support this claim improvement of drought tolerance. We agreed that it had to be field trials. Some people were proposing to make pot experiments and measuring the accumulation of proline. Proline is a physiological indicator of. Yes, but it will never be enough, okay? So I think it's important to reflect on what kind of data we require, but at the end, as I said in my last slide, the proof of the pudding is in the eating. Yeah?
Okay. But have you done any like field trial with commercial
Yes.
products?
We had a project in Belgium where we compared laboratory essays, greenhouse trials, and field experiments with selected commercial products. We want the idea was to see, okay, could we have some laboratory assays that are good predictors of what we could observe in the field, etc. Well, to be honest, it did not work well. Maybe we couldn't anticipate the answer, but it's really impossible to correlate the data sets of what you have in the lab and what you have in the field. Okay, but we wanted to do the analysis to have. And in that case, we decided, because we were hesitant, should we work on specific seaweeds, amino acids, or whatever, or we take commercial products. So we decided to take commercial products. We had 3 years on-farm experiments, And well, it was quite difficult over the years because also conditions were very changing from one year to the next, etc. So to be honest, we invested lots of efforts to universities, one company, etc. And after up to 4 years of research, I cannot tell you today, and it was a bit frustrating to the company because they were expecting, okay, after 4 years, you will tell us we can do this and that test, and it will be a good predictive test for nitrogen uptake efficiency or drought tolerance. And to be honest, we could not fish that out from after 4 years of experiment. So it's still, it's still difficult. I know what I want.
Yo te hago una, pero lo traduciste a Fer vos, porque si no, no me va a entender.
Para eso.
Me va a entender, pero no.
Sí, dale.
Prefiero no.
Sí, dale.
No.
Que no me entienda a mí.
Sí, vos te entiendes porque en inglés hablas más lento que en cordobés. Dale. Te va a entender. Che, no. Mi pregunta es si realmente las empresas, ¿no? Las different companies, están interesadas en entender los mecanismos. O en realidad lo único que les interesa es porque uno tiene otro costo de investigaciones.
He's asking if the companies are interested in knowing the mechanism behind the processes because it's a lot of funding for this.
Y probablemente quedan, no, hagamos franjas con y sin.
Okay. Let's make something simple without
Y damos rendimiento.
only yield, only measuring yield. This is maybe because of the cost of the investigation.
Yeah, that's right, but they should also be aware that there are so many people selling seaweed extract. So if you know the mechanisms and the bioactive compounds, first it's important for quality control for the manufacturing process. If you don't know exactly what are the main bioactives, how can you really check the quality of your extract, etc. Well, that's important, and also to provide the right advice to the grower. It is also important to design the trials. A difficulty with biostimulants is also in intellectual property, how to protect, because many seaweeds are on the market. So we have patents on the manufacturing process, but also the knowledge on the mechanism of action will be a way to be competitive and maybe to not to share too much of it, but that will be a way maybe to design patents on specific applications. But then you need to know more about the action mechanism. For instance, you can patent if you have a technology allowing you to retard the pot shattering, the opening of the pots. I have in mind one example. It's on oilseed rape, but you have the same in soybean, which you have to avoid the early opening of the pots because you lose seeds and you lose yields. So you can design a technology, and there is a clear application of seaweed extracts which have oxinic activity, hormonal activity, which retards the opening of the pot. So one way for the company is I have a technical problem to solve, I have a technical solution, it's innovative, and I patent the application. Okay, that's another way, I think, for companies to be motivated to know enough about the action mechanisms, because it will open possibilities for intellectual property protection.
Respecto a la fitotoxicidad de herbicidas, en Argentina tenemos mucho problema.
Yeah, that I heard about that. That's very interesting. In Europe, the problem would be that we have under the plant protection product regulation, which is different from the fertilizing products regulation, and the placing on the market along that route is much, the PPP route is much longer and complicated and costly. We have what we call herbicide safeners, products that we apply for helping the plant to withstand the herbicide treatment. So there's a category there in the PPP regulation, which means that if you use a biostimulant as a herbicide safener, probably it will be assigned to the pesticide regulation.
It happened the
So
same here.
that's why I already discussed with companies. I think there's something interesting there. No, but we will not be able to place this on the market as a biostimulant. It will be a safener, and it will be too much money for us.
It's a shortcut.
Yeah, but in Argentina, I understand it's not the case, because in the report which you give to me to this morning, I think how was it named, the report on the specialty meta-analysis of fertilizers, in Spanish. Okay, you mentioned the different plant biostimulants that you were discussing, and hormones were included. Huh? In Europe, again, hormones would not be included because they have been placed on the market since years as plant protection product, and they will stay there. Although it's a nonsense, because you can have the same effect by applying synthetic cytokinin, for instance, and the same effect by applying a seaweed extract naturally containing cytokinins. So you could have the same agronomic target, different products, and at the end it will be completely regulated in completely different ways.
No, it's different regulations. This regulation takes a lot of time of discussion, how to classify, and with this kind of products, there are things that have the same action for different. That happens here.
Pero yo no sé, pero acá en Argentina no hay estándares como comentaba él el otro día para la clasificación.
No, there is a standard. Senasa has a standard.
Pero para poder escribirlo tiene que tener eso.
Sí. The definition, the definition I make of inoculants was the last definition of inoculant from Senasa. Senasa is the organization that regulate these products. So they have a lot of definitions. The bioinsumos and all this stuff, they have different definitions and different classifications based on Brazil, based on Euro, based.
More easy than... Es más fácil inscribir un bioestimulante que un protector o regulador. Entonces mucho se inscribe como bioestimulante, aunque tenga...
Sí, entran todos ahí, por eso.
Entran todos ahí, por eso en bioestimulante hay muchas cosas mezcladas.
Además hay fertilizantes que están...
Sí.
I have a question. I have read that I have been working with the group of Rotham, working with the sugar that made, but not all the results. But I read the regulation of the European Union of how the statistical analysis they made with and the p-values they use. What do you think? They use p-values of 0.15, for example, for accepting and explaining as valid. 15.
Yeah,
An opinion.
I was not in favor of that. I don't see why you should use less stringent statistical requirements for certain products and not for others. To be honest, I'm not a statistician, but
they have an explanation of why they use that. It's very weak.
Yeah, but
it's very weak. They say if I have five. The explanation is if I have five experiments with 0.15 by multiplying the probability, you have less than 10. But each one is not significant. So,
but for me, one of the big problems is that we have requirements of field trials, but we have no mandatory pre-registration of the trials, which means that a company can make a cherry picking of the best trials.
Yeah, sure.
Okay, just give 3 trials with positive results. Okay, they make 50 trials. They take the 3 best ones.
5.
If you remember the slide I showed you this morning with such huge variation. Okay, I take the 3 best ones. So this is for me a bigger issue. Yeah, you know, I was invited by the Belgian Ministry of Agriculture to join a technical committee of the European Standardization Organization to work on the standards. Okay, fine, that's not what we need to implement the regulation. I registered to this organization and I was asked to pay a fee. I was saying, I should pay a fee? I'm the expert, you should pay me. I don't want to be paid, but you should pay me. I should not pay.
to participate in the group.
I understood that, in fact, when for the Ministry of Agriculture obtained a seat for me without any payment, okay, fine. And when I arrived there, I understood why. I was the only guy from the academia. All experts were from companies. And so what was happening is that, okay, if you think about companies, they fix the standards for the placing on their markets of their own products.
Yes.
Okay, I think that they want to make also safe products to put on the market. Their motivation to be there is to have products which are safe and which provide added value to the grower. So there's a convergence of interest, I would say. We work together into the same direction, because what they want also, those guys from those companies, we don't want all these snake oils, all these products who spoil the reputation of biostimulants. And so basically they were there with a positive mind. But still, at a certain time, you feel like for the statistical or for the choice of the trials, and there are certain things which were a bit difficult to follow.
Yeah. Parts of the market.
Between us, this is between.
I have a question. Maybe it's a little out of the topic, but and also for both of you. So one time I was in a speech from a pesticide company that is one of the big ones in the world, and they sell a fungicide for rice. Is I don't know, the molecule is isoprotiolan in Spanish. But they say in their trials the fungicide control the disease, right? But when you compare two crops that were healthy and you apply the fungicide, they claim that the fungicide also increased yields. Yeah. No, but a healthy ones, and they say that is because of the composition of the fungicide. And I asked them, they were Japanese, and they have a complete research center, and they discovered that. And I asked for the papers. Of course, they did not have any paper, but I was thinking, how could a fungicide increase yield? And I don't know.
We should see which molecule it is, and there might be many reasons for that. It's not impossible. Huh? It's not. No, okay. You have not to rule it out. Yeah.
El simposio pasado vino un brasilero que habló un poco de esto y que muchos fungicidas tienen promoción de crecimiento.
Okay.
Y por eso dan más rendimiento sin que haya enfermedades.
In Uruguay, with the rice, happens the same. They accidentally discovered that, I guess it's thiamethoxam or some molecule as an insecticide. They use it for an ear in particular that they were expecting problems with that pest, and they discovered that it promotes rice.
Yeah, I've heard about that. Yes.
Yeah.
Yes.
And now they are using it like extensively and won't replace it because not because the pesticide effect, but the plant growth.
Yeah, but it was registered already as a pesticide. Yeah.
They discovered it accidentally.
Accidentally, yeah. Because in Europe, if something has been registered as a pesticide, there's no way you will not be able to move it to the biostimulant.
Here, regulation is the same. You cannot use a product for other things that is not. This is the law. And everybody can do what they want. But.
Patrick, I have another question. After making research on this topic, could you say that there's like a promising classification of the biostimulants, like humic acids for this particular function, amino acids for that another one?
Okay,
you almost answered it, and you said that it's very case to case, but
yeah,
like something in general.
Well, some trends, maybe if you want to work on nutrient use efficiency, routing, I think that humic acids tend to be better than seaweed extract. Seaweed extract may be more effective for drought or abiotic stress tolerance. There is some trend, but you can, for each category, you can find papers showing effects on many things. Amino acids, protein hydrolysates, tend also to be able to modulate nitrogen metabolism, which makes sense because you supply amino acids. But it's again, it's very difficult to generalize. But yes, if you are willing to promote rooting of young seedlings of maize, go to humic acids more than seaweed extracts, for instance. The literature tells you that okay, it's more effective with this humic and fulvic acids than with others. But again, difficult to generalize. Yeah.
Right. And I don't know if I have another question regarding the what Fernando said about the p-values.
Yeah.
Yeah, I'm in favor of using like a 0.05 p-value, but I understand why they use 0.1. As we have seen, like in those meta-analyses, the response to biostimulants is usually around 5%, 7, 3, depending on the case. So maybe the spatial variability of the field where you perform the trials, it's higher than that. So I work for a company in Uruguay that it's starting to make the registers of the products and that stuff. Mm-hmm. I always working with people who makes the field trials, and they told me that they use less strict, in some cases they use less strict analysis, because if not, like most of the products won't be registered.
Yeah.
I know, I understand, because I also work with the statistics and I'm in favor of the lower p-values, but I don't know. That's
I have my opinion. You can go. You can know how strict to the 0.05. You can move between the 0.10, but 0.15 is very low. You can go say the trend, maybe some part of the error. You can move. This is the where you can move easily, but below 10 is like very.
I would say moving from 5 to 10.
From
not further.
No, below is for me. No. When you know statistics, you know what it represents. It's a lot. Maybe we were discussing here. I say maybe what you have to do is how you design. How you design the experiments. Sometimes the experiments are not well designed for testing this. Maybe you can increase the number of size, the number of experiments to have a larger N. Or in the case of biological, sometimes they're claiming that they have more nitrogen, something like this, and sometimes the protocol is the bacteria, and the bacteria minus 20 kilograms of nitrogen. I don't know, because they believe that the 20, so maybe the design has to be a crop response curve with and without, and to compare the slopes or something like this. And this is the way to catch a very small difference that you have with all these products. That's my experience.
I have one question for you.
Okay.
Ayúdame a traducirles, así no se queda afuera. No, yo me quedé pensando, vos mostraste lo de solubilizadoras de fósforo, ¿no? Y eso, pues yo trabajo mucho en micro, entonces siempre te dicen, en la Petri dish está todo divino, después cuando van al campo no se ve nada. Si tenés idea si trabajaron con bacterias o buscar bacterias que sean o hiperliberadoras de, no sé, lo que sea que liberan para o sea, algunas son por ácidos o enzimas, qué sé yo, o directamente tirar el producto final, porque muchas veces lo que pasa en el campo es que hay una inhibición por producto en en la síntesis de esas hormonas o lo que liberan las bacterias, que, o sea, si vos por algún lado lo noqueás, como que seguiría continuamente, o sea, no se enteraría que ya tiene, claro, que ya tiene que dejar de liberar lo que sea. Pobre.
He's asking me about the solubilizing bacteria, and he's asking me because, if I understood good, is that you have sometimes bacteria that produce a lot of acid phosphatase, and they don't know that they have to stop producing something like this. What I see. I think that what I try to make here in the speech is to show that we don't have evidence more than doesn't work. So we don't have evidence that the bacteria is giving the phosphorus to the plant. This is not. There are a lot of research. There are a lot of research of classifying the bacteria. The bacteria solubilize the phosphorus in but when you go, you don't know if this phosphorus goes to the plant. That is happening when the rhizobia. You know that the nitrogen goes to the plant. So maybe the model should be different. So thinking about this bacteria in 10 years, you are applying, and you have to test if the phosphorus in the soil is increasing because of using this technology. But there is a question mark. There is no true.
El otro problema es que, aunque pongamos el fósforo solubilizado, que yo cuando a veces estudio el fósforo, podés poner el fósforo líquido, o sea, y la planta toma solo una parte, porque el suelo...
Pero toma fósforo de la solución, o sea, no
toma
sabe de
fósforo,
dónde viene.
Claro, pero si una bacteria va a solubilizar una pequeña cantidad...
Ni se entera.
Siempre está el resto del hierro, el aluminio, todo eso que te lo va a tomar. La competencia con la planta es muy fuerte.
That I miss is because for marketing purposes, they were selling the solubilizing bacteria as a replacement of the phosphate fertilizer. And that was a mistake from the commercial point of view, because it's not the replacement. It's just a bacteria that solubilizes phosphorus for it to grow and live below. But it was a nice marketing.
No, iba a preguntar relacionado a eso, o sea, por ejemplo, pues pasando ya a mi zona, en una zona que tú conoces allá, tan ácida, usar esos solubilizadores de fósforo, cierto que no, y además no lo dejaría más disponible para...
Claro, it's worse because you have the acid soil with the aluminum.
Lo que pasa es que la planta no sabe, no dice el fósforo este viene del solubilizador, este fósforo viene del suelo, pero está bien, pero cuando vos pones cualquier fósforo, líquido que sea, en realidad eso pasa a la solución del suelo o interactúa con la matriz, entonces es más importante las propiedades del suelo que...
Pero viste que están en equilibrio.
Sí.
Si vos desplazás un poco el equilibrio hacia la parte donde está más soluble y...
Pero es muy difícil porque...
Para mí son hipótesis.
0,01 ppm de fósforo. Entonces,
podemos hacer esa cuenta los pibes en la materia.
no, parece es muy difícil.
No,
Sí, yo entiendo, pero...
I think that we need more evidence. Maybe it's a hypothesis, but you have to test it.
Por ahí tiene más sentido esos otros bloqueadores de los lugares, de los sitios que tampoco funcionan, ¿no? Que bloquean el calcio, el aluminio y el hierro, entonces lo que parece, por lo menos no se lo come la tierra.
Sí. ¿Cómo se llamaba el producto ese? Era el Avail. Era un producto, un protector.
Y esos fertilizantes, por ejemplo, fosfatados que vienen con supuesta tecnología que más calcio, ¿qué hace?
Lindo dibujito. Así.
Linda historia.
Sí, me parece que es poquito, nada, es como...
¿Y qué tal? Perdón, a mí es algo del tema, pero viste que algunos vienen, por ejemplo, superfosfato triple, roca fosfórica acidulada y otro. ¿Sí? O...
En cuanto a solubilidad.
Y en cuanto a un suelo fijador, que sí pueda mantener eso, o sea, que...
Las rocas fosfóricas...
Es como uno de liberación lenta la roca fosfórica, en ese caso.
Es el único.
Claro, pues digamos mi pregunta, si yo aplico, por ejemplo, solo superfosfato triple... Si yo aplico solo superfosfato triple, un ejemplo, y lo comparo con roca acidulada, ¿sí debería haber una diferencia entre fuentes? O sea, dentro de lo teórico, en un suelo muy fijador, ya sea por calcio o por aluminio.
En suelos ácidos fijadores podría ser bueno.
Ok.
En los neutros de acá,
Okay.
Siempre mejor el soluble.
Ok, listo. Bien.
Es como que de golpe le da un chorro de fosfato y lo va a tomar.
Ok.
Y siempre que sean fijadores, no quiere decir que sea solo rojo va a ser fijador. Porque muchas veces solo rojo no tiene aluminio.
Ah, bueno, sí, 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 sí,
Entonces,
sí, sí, sí.
Es otro error, digamos.
Sí, 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 es verdad. Sí, claro.
Ver rojo y decirte fija fósforo, no necesariamente.
Oh, y hay suelo negro que fija fósforo, por ejemplo, los antisoles. Nosotros tenemos un montón de problemas con eso. Y
O suelos con
Siempre uso
calcario.
del paper de ustedes, es buenísimo, la absorción.
O suelos calcáreos, que vos tenés suelos calcáreos, también tenés problemas.
incluso se me hace más difícil manejar un calcáreo que un ácido. Que un ácido al final en calas, pues un ácido rojo en oxisol en calas y lo solucionás, ¿no? Más o menos.
Sí, pero la ligación con el calcio es más fácil de romper
¿Sí?
que con la del aluminio y el hierro.
Ok.
Lo más soluble.
Ah, bueno. Sí, sí.
Bueno,
Bien,
armamos.
Un pequeño
¿Alguna pregunta más acá?
¿Vamos? ¿Vamos?
Muchas gracias.
Gracias.
Thank you. Thank you, Cesar, for this.